Hello everyone, a very good evening to all of you. So I hope so you are able to visualize the screen and you are able to hear me as well. Can you please confirm in the chat? Okay. See, cell organelles and protein targeting is one of the important topic, as I told you from the very beginning itself, right? So the thing is that the questions do come from these topics a lot. So that's the reason most of the weeks, like last week also, I have continued with cell organelles and protein targeting. This week also, the same thing, because protein targeting is a topic where you get a very good number of 4 marks questions in every CSAR exam. Okay, so good evening, sunshine. So let's have a look into the first question of today's session. A null mutation is created in a gene which is responsible for specific phosphorylation at sixth carbon position of mannose on acid hydrolases occurring in cis-Golgi network. So these are the statements that is provided to explain the effect of the mutation. The lysosomes will be devoid of lysosomal enzymes. Lysosomal enzymes will be secreted out, or the lysosomal enzyme will get localized in the cytoplasm. Now, here are the options for you. You have to identify the correct combination of statements that explains the effect of mutation if acid hydrolases in the mutant will not be degraded. So these are the options for you. Let me see how many of you are able to give the answers, and these are the statements. Lysosomes will be devoid of lysosomal enzymes. Lysosomal enzyme will be secreted out, or it will be localized in the cytoplasm. So which combination, according to you, will be the correct one? Can anyone tell? See, here just try to remember the protein targeting that takes place within the lysosome. How the signal sequences are attached and how they are traveling into the lysosome. If you remember that image, things will be more clear to you. So is there anyone who could give the answer of this question? Most of the times when you look into this protein targeting, majority of times you get direct questions and sometimes you can expect this mutation related questions. Quite a tricky one, but still if your basic concepts are clear, then you'll be able to attend the session. Okay, I can see Anupam has given the answer. What about others? Lysosomes will be there. So what is the consequence of mutation? That's what they are trying to say. And these are the combinations. 1 and 3, 2 and 3, 3 only, 1 and 2. What should be the correct combination for the statement? Yes? Can anyone could answer? This is a very easy question. Although it's a 4 marks question, just give a twist to your thinking. Okay, I can see Priyanka, Sandhya giving the answers. Anyone else? Anyone else would like to give your inputs? Okay. See, the correct answer over here is option or statements 1 and 2. Now, how the statements 1 and 2 are correct? See. Statement 1 indicates what? The lysosomes will be devoid of lysosomal enzymes. So, why do you think this one will be the correct one of the correct combination? The first thing is that we are aware that, so what is the signal sequence for entering into the lysosomal compartment? Can anyone say? Good evening, Anusha. Can anyone say what is the signal sequence to enter inside the lysosome? Like every organelles, you are having a signal sequence because without a address, how a protein will enter inside the target location? Am I correct? So, like that, what should be the signal sequence for any protein to enter inside the lysosome? Absolutely correct. Mannose 6 phosphate, right? So, if that itself is mutated, how you can say that the enzymes will be targeted to lysosomes, right? So, that should be the first answer. And what was the other combination? Answer option 2. I mean, the statement 2. Lysosomal enzymes will be secreted out. So, anyone remembers which organelles are involved in the lysosomal transportation? Once a protein for lysosome is synthesized, it goes to which organelle, and from which organelle it goes into the lysosome. You have got a mediator organelle, if you are aware. What is that organelle? It is your cis Golgi network, right? So that is the answer. If the mannose group is not phosphorylated at sixth carbon residue, then the lysosomal enzymes will not be targeted to lysosomes because it is not having that sorting signal. And since lysosomes are devoid of lysosomal enzymes and they will be present in the Golgi, so from the Golgi, what happens? Usually the proteins will be secreted out of the cell. If you remember, last time I had a question, I had taken a question with you regarding the fluorescence protein. So, what was the location? From rough endoplasmic reticulum, the proteins will be carried with vesicles, that is, transport vesicles, into the Golgi compartment. And from the Golgi compartment, it will be secreted out. So, how it will be secreted out? With the help of, again, vesicles. So, that's why the statements 1 and 2 are the correct. Okay. Next, identify the correct combination over here. That means on the column B, you are provided with signal sequences, and column A is provided with cell organelles. This was again a 4 marks question. I think 2 years back they had asked this question. And these kind of match the following kind of questions are always expected. And if they're provided with match the following, never skip this kind of questions because you can do it. Because anyways, out of the 4 combinations, you will be sure of at least 2 or 3. So you have to find the other one. Can anyone give the answer for this question? Okay, I can see the few answers came. Surender, Suresh, Sunshine, Anusha, Gayatri. Okay, anyone else? Anyone else would like to put your inputs over here? See, some kind of match the following kind of questions. What happens, if you get one combination correct, you will be able to get the entire 4 marks if you are lucky enough. Okay. Okay, you are quite sure for lysosome and nucleus. C. So in that case, you can get the answer. Okay, let's find out the answer. The correct answer over here is answer 3. Now, how we can say that this option is correct? First thing is that blindly we can say the only organelle which does not have amino acid as their signaling sequence is lysosome. So A to 4, you can easily connect, right? So when you got a confidence that A to 4 is correct, obviously you don't have to look for options 1 and 2. Next, what you need to find? B to 3. So B to 3. That also may be quite confusing because again lysine and arginine residue is there for mitochondria as well as for nucleus. But again, you can pick up another thing. What is that? Peroxisomal sequence. Anyone remembers what is the signal sequence for peroxisome? It is mentioned as C-terminal tripeptide. Does anyone remember what is the signal sequence for peroxisome? The way you said me, a lysosome is mannose 6 phosphate. Peroxisome, what should be this tripeptide? What does this tripeptide indicates? Absolutely correct, Suresh. Anusha, yes, the sequence will be SKL. Instead of this, if they have mentioned a 9 terminus, N terminus 9 amino acid residue, then also you can easily click on to peroxisome. So this sequence is otherwise known as PTS1 sequence, and this sequence is otherwise known as PTS2 sequence. Okay. Now come to the difference between nucleus and mitochondria. No doubt, both of the motif which we can say that is required for entry into the specific organelle, we have lysine and arginine residue. But what is the difference is that always remember for nucleus, it is not specified whether it is N terminus or C terminus, but for mitochondria, it is always N terminus. Okay. So with that option, you can easily identify the difference between a sorting signal moving to the nucleus to that of a mitochondria. So in case if you have any confusion in the combinations, try to eliminate the options with one correct combination. Then things will be all easy for you. See, if you are confident with this, you can easily eliminate this. Now you have to find what is the correct among the two options. Right, so that will be easy for you. So try to do in this way. SKL stands for amino acid sequence. Serine, lysine, leucine. That is the conserved amino acid sequence, Anupam. But for N terminus, that 9 amino acid sequence is not yet deduced what it is. Okay. Moving on to the next question before going to that. So here, this is a table that provides you with different types of organelles and what is the nature of the sequences. You can easily find in Lodish book. And another thing to be considered over here is, the pre-sequence, that is the signal sequence. In case of peroxisome and nucleus, it is not degraded. But in case of other organelles like ER, mitochondria, and chloroplast, upon reaching the destination, the signal sequences will be clear. This one point you can consider. Okay. Then another table which I have, it's being taken from Albert's, and in this case, the sequences are mentioned. Well, as such, these long sequences, they are not going to ask, but yes, I think last to last year, we had a question directly. This sequence was asked, and this sequence was given in the question, and they had asked where it comes, like where the protein containing this signal sequence will be going to. So that was a kind of direct question. Okay. But I don't think so. This much long sequences you have to remember. Just remember what is it rich with. Like here it is mentioned, right? If it is endoplasmic reticulum, then it should be rich with one or more basic amino acid residues. Then here also the same thing, but along with that hydrophobic residues will be present for mitochondria. Chloroplast specific motif is not there, but it should be rich with serine threonine residues. And peroxisome, too, we have seen PTS1 and PTS2. Nucleus also, I said you, it is lysine to arginine as a common residue, but it does not have any N or C terminus. That is the difference. Next, moving on to the next question. Which one of the following proteins is most likely to be found in the intermembrane space of mitochondria? A protein that contains N-terminal matrix targeting sequence followed by hydrophobic stop transfer anchoring sequence. Second, again MTS, that is matrix targeting sequence followed by cleavable hydrophobic sequence that blocks the complete translocation. Next, a protein with multiple internal sequences being recognized by TIM22 and a protein with an outer membrane localization signal followed by an MTS signal. So what should be the answer over here? Actually, in my last session, I had given you the signal sequences of all the compartments. Compartments in the sense, mitochondrial compartments, like what is the sequence for entering into intermembrane space, then inner mitochondrial membrane, then matrix. So accordingly, what should be the answer over here? You have to identify a protein that needs to be targeted to intermembrane space of mitochondria. Remember this word, intermembrane space. Try to eliminate the options and get the answer. Try to eliminate the option and get the answer. I can see majority of you are saying as option 3. Read the options properly. N-terminal MTS followed by stop transfer anchoring sequence or followed by cleavable sequences, then internal sequences, then your outer membrane sequences. What should be the correct answer over here? For this, you have to remember the images which I had shared with you last time. Every protein having a specific sequence to enter inside a specific compartment. Anyone is giving the answer? Anyone else would like to give the answer? Sometimes these kind of questions, they will provide you with images. Like the last time in the last session, I had asked you, if you remember, I had provided you the images and you have to identify where that protein goes. Sometimes they will ask in this statement based questions. Option 1 may be. So you are having confusion. Sunshine giving as option 4. Do you find it difficult to solve these kind of questions? Well, if you are able to remember the sequences, the nature of proteins, these kind of questions you can easily pick up. And remember, these kind of questions usually comes in the form of 4 marks. So easily you can fetch a 4 marks from here. Okay, let me give you the answer. The correct answer over here is answer 2. That is an N-terminal matrix targeting sequence. So they have mentioned matrix targeting sequence is present, but it is followed by cleavable hydrophobic sequence. So that particular sequence is cleaved so that complete translocation is prevented. This you cannot say, because here regarding the cleavage is not mentioned, and this is also not possible. Multiple internal sequences being recognized by TIM22 directly. TIM usually comes where? In the inner mitochondrial membrane, right? So there is no such protein which will directly recognize the inner membrane protein, like the TIM22, like that. But it can recognize the outer translocon channel. Inner translocon, it is difficult. Then if it enters into the outer membrane translocalization signal, then how it will enter into the MTS? And if it enters into the MTS, you don't have a specific sequence that will help it to tether to the intermembrane space. So that's why you cannot opt for option 4 as well. See, if you remember, I had discussed with you in the class regarding this. This one does not require any kind of receptors or a channel specific like TOM22 or TOM20, but directly translocon is required. It does not require either the inner mitochondrial membrane exposure. So in that case, it will enter into the translocon channel, released into the intermembrane space. And if this protein is disulfide bonded, they are the one who generates the disulfide bonds and incorporates it. But if you look into this image, this protein is having the MTS, which was there in our question. That means there should be some exposure to the matrix, because without the use of exposure to the matrix, there is no requirement of tagging MTS over here. Because if you see here, you don't have that MTS. So that means they are not even exposed. A small part is also not exposed. But in this case, you are having, that means it has to reach the matrix. So after reaching the matrix, you will be having the matrix processing enzymes, which will cleave away the sequence. Then with the help of lateral diffusion, it will be released into the inner membrane. And since it is having the hydrophobic cleavable sequence, this will be cleaved and released into the intermembrane space. where it will be sequestered with the protein known as heme, remains folded properly. So this is how you need to attend it. Okay. Next, in regulating the quantity of enzymes, its degradation plays a pivotal role. So, following statements are made to represent the degradation of enzymes in 26S proteasomes. So, if you are aware, many a times you come across a stage where the proteins needs to be degraded. So, if you want to degrade a protein, what is the first signal that should come? Do you have any specific tags over there that will lead it to proteasomal assembly? Yes. Heat shock proteins will be there, Anupam. Yes. Do we have any specific tags that will be helping it to target unwanted proteins to proteasome? Very good. It is your ubiquitin tag. And do you know at which amino acid this ubiquitin tags will be given? Yeah, you have to focus on that word, Anchal, as you mentioned, cleavable. That word is mentioned now, so you can easily get your answer. I mean, you can differentiate the options. Yes, absolutely correct, Anupam. It will be your lysine residues. Okay. So many a times our proteins has to be targeted to the proteasomal assembly. But basically we don't study much regarding the proteasome. But even if you don't study much regarding this ubiquitin tags and proteasome, still you will be able to answer the question. See the question? The active sites of proteolytic subunits face exterior of the proteasome cylinder. Second statement, face interior of the proteasome cylinder. Third statement, degrading enzymes will be targeted to the exterior of proteasome by covalent attachment of one or more subunits of ubiquitin. Or degrading enzymes will be targeted to the interior of proteasomes by covalent attachment of subunits. Now you have to find out the correct combination over here. Can you find out the correct combination over here? So here you don't have to understand what is the structure of proteasome, but if you have a basic idea that proteasome leads to the degradation of a protein, that is breaking them into monomeric units, you can easily get the answer. Okay, I can see the answer coming up from Gayatri. Any other answers? Okay, Sunshine. Suresh, I guess you are able to answer this question, right? Even though the entire thing regarding how this ubiquitin tags are added, to which amino acid it is being added, and how the proteasomal cylinder works, in spite of that, you are able to give the answer, I guess. Okay, I got 3 answers so far. Any other answers? Okay, fine. Deepshika, Anupam. Anyone else would like to put your answers here? If you just simply read the options carefully, you will get the answers. There is no toughness in this question. In fact, this is the question being asked in the last exam. I don't remember whether it is morning batch or afternoon, but it came last year only. Exactly, Sunshine. So, shall I give the answers? No one else would like to add on? Anchal, would you like to try? Would you like to try on, Anchal? Others? Okay, fine. See, the correct combination over here is answer 3. I can see many of you has given this combination, that is B and D. See, the active sites of proteolytic subunits faces interior of the proteasome cylinder. So if this is correct, in that case, degradation will also occur within the interior of proteasome. And covalent attachment, so I have already said you, it will be attached to the specific amino acids with the help of ubiquitin. So ubiquitin tags will be given, then only it will lead to proteasome. So if you look into the image over here, this is what, or this is how it looks like. So this is your proteasomal assembly with a cap in the top portion and in the bottom portion. Because from the top, your proteins will enter inside. Here the processings will take place and from here it will be released outside. That's the reason you are having these two caps. Then you are having this ubiquitin tags and this is your target protein. Remember, there are two types of subunits present within the proteasome, that is alpha subunit and beta subunit. Okay, so you can see the arrangement over here. So ubiquitin tags protein will be taken into the proteasomal assembly, and within that you can see that beta subunit is responsible to carry out the catalysis. Okay, so it has taken up inside, catalysis is being carried out, and finally the particular protein is broken down into monomeric units, which can be utilized by the cell again. So this is how your proteasomal assembly occurs. Okay, so clear to everyone? I hope so. No confusion further. Anupam, it's clear, right? So moving on to the next question. Following are the statements made about Kedal receptor. Are you aware of Kedal receptors? Are you aware of Kedal receptors? Kedal sequences, Kedal receptors, which organelle it will come for? Absolutely correct answer. Yes, it's correct. It's your endoplasmic reticulum. So somewhere it is related to ER only. So let's check out the statements. Kedal receptor is found in COP1 vesicles. Kedal receptor is having affinity for Kedal sequences, that is higher at high pH. Synthesized, the receptor is synthesized on the ER, or the receptor can present both in the ER and Golgi compartment. So you have to identify the correct combination within the Kedal receptor. I mean the statements based on the Kedal receptors. Can anyone identify? Read the statements carefully. You'll get the answer. So COP1 vesicles, COP2 vesicles, all those things are being mentioned. You have to have a clear-cut idea about the Kedal receptor first, significance of Kedal sequences. Third, the difference between COP1 and COP2 vesicles and how the transportation takes place. If you are able to understand that, you will be able to give the answer. Just find out. Okay, I can see Anchal is giving the answer. Okay. Do you remember the difference between the COP1 vesicles, COP2 vesicles, which I had taken in the class? Okay, I can see a combination of answers coming up, either 4 or 3, that combinations are coming. Okay, COP1 transport to the nucleus, is it correct? Just have a think once again, because mainly your vesicles, whether it is your COP1 or your COP2 vesicles, they are related with ER and Golgi. Am I correct? Anyone else would like to give the answer? Yeah, it will not come as nucleus. I think you got some, you forget maybe, Sunshine. Yeah, Golgi to ER will be the correct. Yes. So the correct answer over here will be the option 3, A, C, and D. So Kedal receptor is found in COP1 vesicles. Kedal receptor will be synthesized on the ER and they can be recycled between these two compartments, that is ER and Golgi. Okay, so let's check it out. How does it works? So this is the sequences. So this is your endoplasmic reticulum that contains Kedal sequences. Okay, and the violet colored proteins which you see over here, these are the ER resident proteins that contains Kedal sequences. So we know that origin of COP2 vesicles will be from endoplasmic reticulum and origin of COP1 vesicles will be from Golgi complex. So what is the third form of vesicle? What is the third form of vesicle? Anyone remembers? We have COP2 vesicle, COP1 vesicle, then we have another form of vesicle. Three types. Absolutely correct. Anchal. Yes, Gayatri, Anusha. Yes, it is a clathrin coated vesicles. And what is the origin of clathrin coated vesicles? What is the origin of clathrin coated vesicles? COP2 originates from endoplasmic reticulum. COP1 originates from Golgi network. What is about clathrin vesicles? What is its origin? Is it from Golgi or from any other compartment? Yes, you are correct, Anusha. Yeah. Trans Golgi network. Apart from that, do we have any other part from where this clathrin can be taken? Yeah, that is the structure which you said me. Absolutely correct, Anupam, which is made up of your triskelion structure with an adapted protein. Yeah, that is also correct. Yes, very good, Suresh. We are having plasma membrane. Okay, so trans Golgi network and plasma membrane is the origin. Now, how the Kedal receptors work? So, these is your endoplasmic reticulum. By mistakenly, the COP2 vesicles has carried this Kedal containing sequences, like Kedal sequences containing proteins, which is supposed to be retained within the ER only. But we don't have a problem because we are having Kedal receptors present. And Kedal receptors are present where in the cis-Golgi network. So within the cis-Golgi network, what you are having, the receptors will carry those proteins that contain Kedal sequences. And we know that origin of COP1 vesicles is from cis-Golgi network. So via COP1 vesicles, the Kedal sequences containing proteins will be carried by the receptor and taken back to the endoplasmic reticulum. Okay, so this is how it works. COP2 vesicles by mistakenly taken up, proceeded ahead, but no problem. Golgi is having the Kedal receptors which carry the Kedal sequences containing proteins and will be carried to the ER back. Is it clear to everyone? This particular thing is clear to everyone. This image is clear to everyone. And another question over here, we know that Kedal sequence is specifically for endoplasmic reticulum, right? Do we have any other sequences? Do we have any other sequences that can target a protein to endoplasmic reticulum? You have a table, Anupam. I think it is there in the Lodish, I guess. In the Lodish, you will find a table where you will find the comparison between COP1, COP2, and clathrin. Okay. So tell me, what is the another sequence for a protein to enter inside the protein? So this is specifically for soluble proteins. Soluble proteins. But if I speak of membrane proteins, no. SRP will be the receptors, right? Signal recognition particle that will carry the content. I'm asking the sequence that is responsible for carrying the membrane bound proteins within the endoplasmic reticulum. Any one of you remembers? Anusha, Anchal, Suresh, Sunshine, Anupam. No, Anupam, it is not SRP. SRP is basically a translocone. Translocone, what is its function? To transport a particular protein from one compartment to another compartment. Am I correct? We have studied under endoplasmic reticulum, we have studied under chloroplast as well. That is not the sequence of ER. We have, if I say, maybe you will be able to recollect, KKXX sequence. You are able to recollect? KKXX sequence that is responsive. There K indicates your lysine and X indicates any amino acid. Absolutely correct, Suresh. Okay. So this is the sequence for membrane bound proteins. Now, another statement which we have seen that ACB is correct. So why B is incorrect? Kedal receptor affinity for Kedal sequence is high at higher pH. That means what? Kedal receptors will bind more tightly to its ligand, specifically at low pH. And it is thought that receptor is able to bind Kedal peptides in the cis-Golgi network and release those peptides in the endoplasmic reticulum. The reason is pH of Golgi compared to that of ER. Do you know the pH of Golgi and ER? Do you know the pH of Golgi and ER? Last time we had discussed about intermembrane space, mitochondrial compartment, and all we had discussed, right? What is this pH with respect to Golgi and ER? Anyone aware of this? See, what happens here is, in general, the pH of the Golgi and ER is maintained at a near neutral or slightly acidic level. Okay. So different regions or cisternae is the finger-like projections you find within the Golgi compartments, will have slightly different pH levels, that is decreasing along the cis to trans axis of the Golgi stack. Yes, Suresh, you are somewhat correct. See, yes, Kirtana, yes, Anupam. See, the Golgi stack, which ranges from 6.7, which is present in the cis-Golgi network, to pH 6, which is present in the trans-Golgi network. So, obviously, the medial Golgi network will be somewhere in between these two ranges. So, we can say on an average, the pH will be around 6.5 to 7, making it slightly acidic, not completely, very minor way. So this low pH is essential for functioning of various Golgi enzymes involved in glycosylation and protein modification. The way I said you regarding the Golgi apparatus, even ER also maintains a slightly, not I will say a complete, because a pakka completely acidic pH means it has to go beyond 4, the way which you are having in lysosomes and endosomes. But here it maintains a alkaline pH. So that is also in the range of 7. So remember this pH of individual organelles. Sometimes they may ask in the form of 2 marks, or sometimes like this questions, if it comes 4 marks also you can get. So remember the pH. Next question. Okay, just before moving ahead, Just wanted to remind you regarding the Watson Junoon Scholarship, where you will get 1 year coaching for CSIR NET GATE exams. Then you will have 12 months of access to all the workshops that are being conducted by Biotechnica. Apart from that, hands-on training is provided for art programming. Certification courses are available in your in Biotechnica, so you can choose 4 certification courses out of that. And you are going to get one VIP pass to our CRISPR conference, which will be held in the upcoming times. So if you have anything to be asked regarding the scholarship program, just give a call to 1800 1200 1818. Just a reminder for the scholarship program. Okay. Next. Now, another question with respect to nuclear transport. So, nuclear transport is also a very favorite part when we go for CSIR exam. Okay. So, here are some statements provided. RAN GTP levels are higher in the nucleus than the cytoplasm. Nuclear import receptors can shuttle between the nucleus and cytoplasm. NTF2 can transport RAN GDP into the cytosol, and export of mRNA is not directly dependent on RAN. tRNA and microRNA is mediated by exportins. Now you have to identify the correct combination of statements over here. Just find out, read the statements. If you want, I will show you the options also, but all you need to do is find the correct combination from here. Okay, just check it out. What is the correct answer? For this, you should have a basic idea with respect to nuclear import, which we had already discussed in my previous session. Okay, just have a look to the statements. And if you want to let me know, I will show you the options also. Okay, I can see Anchal is giving the answer. Okay, you feel that A and B are correct. Okay. What about others? Yes, you just share your opinion, because anyways, I'm going to provide you with the correct combination. Okay, I can see many of two of you, Surender and Suresh. A, B, and E is being said. Okay. Kirtana also following the same. Okay. Okay. Anyone would like to further add? So these are the combinations. Okay. A, B, C, D only, B, C, D only, A, B, D, E only, and A, C, E only. This is the combination. Maybe you can focus on the statements and tell which all are correct. So as you mentioned, ABE as a combination is not present. AB as a combination is not present. Either ABCD, BCD, ABDE, that means C is not there, and here B is not there. B and D is not there. So which has to be the correct answer? If you want, I can show you the statements once again. Okay, I can see many of you has opted for option 3. Any other options? Okay, Sandhya. These are the statements. See, this part is very important from examination point of view. Nuclear transport is always an expected question when you go for CSI. Okay, let me give you the answer. The correct answer over here. Okay, I'll come to that answer. Yes, I'll come to that. Answer 3, that is A, B, D, and E. As majority of you has mentioned option 3 as answer, that is absolutely correct. Now, what does this A, B, D, and E indicates? A indicates RAN GTP levels are higher in the nucleus than cytoplasm. Do you remember the enzymes which keeps the RAN GTP at higher level in the nucleus? Do you remember the enzyme which keeps the or which converts the GDP to GTP and keep it at higher concentration within the cell organelle, that is nucleus? Anyone? Now, again, if I will tell you the answer, you'll be able to recollect. Try to recollect. Actually, we are having two enzymes which works in the cytosol and another one which works in the nucleus. So based on that only, you are having GDP and GTP levels are present. Absolutely correct. Anchal, Surendra, Suresh, Sunshine, Kirtana. Yes, yes, yes. So here you are having gap and here you are having the gel. Okay. Next, nuclear import receptors can shuttle between nucleus and receptor. Nucleus and cytoplasm. No, Anupam, the other way round. What you said is the other way round is correct. Jeff for nucleus because they are present in the chromatin. So where should be our chromatin present? It should be present in the nucleus. Am I correct? So, what are these nuclear import receptors? Do you remember? If you remember the nuclear transport as a signal transduction pathway, I mean the protein targeting pathway, you'll be able to remember the receptors. Who are the receptors? Who are the receptors? Importin is fine. Can you name me the importin? Which one? Importin is correct. But along with that, you are having, yes, very good. Importin, alpha, and beta. These are considered to be the receptors for taking a cargo to the nucleus. Then this is incorrect, they said. Okay. This is also export of mRNA is not directly dependent on RAN. This is correct. And tRNA and microRNA's export is mediated by exportins. So let's check it out why C is incorrect. Okay. C. As you told, just keep this image in your mind. RAN Jeff, where it is present in the nucleus. That means tethered to the chromatin. Okay? So here you will be having GDP to GTP exchange. So you are getting the RAN GTP level in the nucleus and gap present in the cytosol keeping the RAN GDP level high. So this concept you are aware. Next, what you are having, why the statement C is incorrect. NTF2. You know what is an NTF2 is nuclear transport factor, which is the transport factor responsible for the import of RAN into the nucleus, not to the cytosol. That is the major setback or the incorrect statement over here. If it was given, see here it is given cytosol, but the correct answer, if it was mentioned as nucleus, the statement would have been correct. So, each importin or exportin mediated transport cycle removes one RAN GTP molecule from the nucleus and releases it in its GDP bound form into the cytoplasm. So these NTRs rely on additional components so that they can refuel the RAN GTP gradient. And what are the components? That is your nuclear transport factors, which can retrieve the RAN GDP back to the nucleus so that within the nucleus you are having the JEF, and that will help in the exchange converting it into the active form. Okay. Now we are aware that mRNA is synthesized and processed in the nucleus. Correct? Transcription takes place where? In the nucleus only. So it will be packaged with some RNA binding proteins, obviously. So what is that known as ribonucleoprotein particles? So despite the complexity in this protein composition, export of majority of mRNAs utilizes a non-karyoferine receptor. Are you aware of karyoferine receptors? Karyoferine receptors. Now, here they have mentioned as non-karyoferine receptor. Are you aware of karyoferine receptors? Anyone? This also we had discussed in the class. In fact, there was a table for that. Karyoferine receptors. Anyone? Anyone? What is that? If you are aware of this karyoferine, you know, then kindly say what it is. Yes. What it is? I can say only, I can see only yes coming up, but I want the answer. What is this karyoferine receptor, sir? See, what is the answer over here is the normal importines and exportines, which we use for importing a particular component inside the nucleus or exporting a component outside the nucleus. We togetherly call it as importine and exportine, which we call it as a karyoferins. Karyoferins functions as a transport and export. Okay, so we have importine, that is the proteins with basic amino acid NLS, and SNR protein, that is SNRNPs, that is U1, U2, U4, and U5. Transportin proteins are basically what? mRNA binding proteins. Okay, and importin 7 is what? Histone H1 ribosomal protein. So these are some examples of import karyoferins. Okay. And export means what? CRM1. That is a protein which is having leucine rich nuclear export signal. So these you cannot expect. But yes, this has just come, right? Exportin T and exportin 5. T is specifically for tRNAs and 5 is specifically for microRNAs. Yes, please proceed with the doubt. Sure, Gayatri. The which one you are asking? You are asking about karyoferins. Are you asking about karyoferins? Karyoferins are nothing, the group of proteins which are responsible for carrying the content inside the nucleus or outside the nucleus. We call it as importins and exportins. So this importins and exportins togetherly we call it as a karyoferin. And examples of importins and exportins, what you are having? See here. These are the examples. Because specific proteins needs to be carried out by specific import receptor. Similarly, specific export proteins needs to be carried out by specific export receptors. Could you get me now? Gayatri? Yes, what is your doubt? If you can elaborate with your doubt. Daisy edits. And Gayatri, is it clear now? Or do you have any further confusion over here? Okay, I hope so you got it right. Okay, fine. So this is the thing. So that's the reason here the statement which is provided. Okay, you got it, right? Very good. So export of mRNA is not directly dependent on RNA, RAN, as I told you. Then tRNA and microRNA, they are being mediated by exportin. So accordingly, you are having the exportin protein for each, right? So that is what I was saying. Okay. Next question. Again, a combination or match the following. Just try to find out the correct combination over here. Try to find out a correct combination over here. Now, this is also quite easy because once you have already solved a match the following question, I guess you are able to solve this one. Okay, I can see Suresh has given the answer. Anupam gave the answer. Others? Okay, Anusha. See, here also you can apply the same logic which I said you. Try to eliminate the options. You will get the answer. Okay, I can see. Wonderful. I can see majority of you are saying the combined answers. And also I had explained you regarding the ER retaining sequences and all, right? So with that also you'll be able to give the answer. Okay, very good. So for the first time I received all the answers, common answers. Yes, Deepshika, Anchal, Gayatri, Sunshine, Surendra, Kirtana, yes. Okay, let me show you the answer over here. As you all mentioned it correctly, and I really happy that all of you has given the correct answers. Yes, the answer is 1. So here also you can apply the same logic. As I already told you, lysosomes is for mannose-6-phosphate receptor. Okay. So this we can easily take as it is. Now we can eliminate second and third options. Right? So there is a tug of war between 1 and 4. So cis Golgi is what, as I told you already, cis Golgi originates from, cis Golgi will be providing us with COP1 vesicles that will drive the contents back to the ER. Right, so B to 3. C to 5, trans Golgi. Trans Golgi is present at the end. So that's the reason the proteins will be secreted out. Right? Then COP2 vesicles, D to 1. Do you remember the difference between anterograde and retrograde transport? Do you remember the difference between anterograde and retrograde? The very common terms, quite similar terms, but there are chances of getting confused. Do you know the difference? What it is? Can anyone tell me? What should be the answer? Anterograde means what? Retrograde means what? Yes, absolutely correct. So anterograde means, remember, moving in the forward direction. Retrograde means moving in the reverse direction. So we have COP2 vesicles that is moving from ER to Golgi. So that is known as anterograde. And this COP1 vesicles, if you take into consideration, it will go on in the reverse orientation, Golgi to ER. So that's why you call it as a reverse. And the last one is E to 2, that is endocytic vesicles. And endocytic vesicles will be your nothing but clathrin coated vesicles. Okay, so that is the thing. And go through the protein targeting specifically. So we will continue protein targeting. Along with that, cytoskeletal proteins also I'll be taking in my next session. So make sure that cytoskeletal proteins, specifically the accessory proteins based questions, are very frequently asked in the exam. So try to study that portion and come to the session so that it will be easy for you to answer the questions. Okay. Okay, then. Thank you so much, everyone, for your answers, for your interaction. See you again in the next session next week. And as I told you, study cytoskeletal system and protein targeting together and come to the session. Okay. Thank you so much.